Hello to everybody. This is Wednesday morning, 10 a.m. So means the family of MD Anderson. It's again all together for another wonderful talk and excellent speaker, Dr. Stephen Lay. So thank you all of you being here with us. I know very well you can choose for very many wonderful webinars, and we really appreciate very much your being so much with us and enjoy science Wednesday after Wednesday. So again, as you know very well, this is organized by Dr. Draeta's office, and I host it with my friend and colleague Alessandra Ferraioli. And in fact, we have to thanks again and again to Liz, Kent, and Laura, Adame, Alison, Garth also, because they are the ones who keep this webinar technically safe and moving forward. And today is my pleasure to introduce you Dr. Stephen Lay. You know very well the one from MD Anderson and the one from abroad, because he's a wonderful clinician scientist and he's very collaborative. It's my pleasure to introduce you, because he's here from the time that I arrived here, and in fact he was one of the first clinician scientists who got in love with what I was already in love in that moment with microRNAs, and we start a very long and fruitful collaboration on understanding why very short RNAs are important in head and neck. So Stephen has a wonderful training. He was trained in UCSF and he was trained also in Stanford. Not many of you know that he is not only a clinician, he is not only a scientist, he is not only a clinician scientist, but he has also a master in economics. So he understands very well how the world is working these days. So he is very efficient, and in fact, you will see, he is a very busy surgeon and clinician, but he is publishing wonderful papers, not only on non-coding RNAs, but also on new technologies who are driven to save patients and to do what everybody at MD Anderson loves to do, to cure cancer. And with this said, thank you very much, Stephen, for being with us. And for you, the attendees, please ask questions, because I'm sure will be a very interesting and innovative talk. Thank you again for being with us.
Thank you, George, for that very kind introduction. It's not often that my economics training is referenced. I would say that even with that background, I'm not sure that much of anyone can explain what's going on in the world these days. But I do appreciate everyone coming today. I'm going to talk about some of the work going on in our lab. And I think with that, the first thing I always like to do is to acknowledge my group. We've all gotten used to kind of the separation with the moat, the isolation, the sort of working remotely and via Zoom and social distancing and masking. And so I just wanted to put up a picture of us from about a little over a year ago when we were able to be all together. I would highlight that a lot of the work I'm going to talk about today has been spearheaded by Nikhil Chari, who's in my group and has been with me for quite some time. And then I'd also like to point out Yunyun Chen and Ying Henderson for all of their strong work. And then Abdullah Mohammed, who has joined us through radiation oncology, first in our imaging work together, and then coming into my lab to do his PhD dissertation work. With all that, before I get on to kind of the work around microRNA and miR-27A star, I would highlight just as much that it was part of George's enthusiasm for microRNA as I was looking at this story that helped to attract me from University of Pittsburgh, where I had done my fellowship and then had started as a junior faculty, but it was really coming to MD Anderson, seeing the opportunities, seeing sort of the sort of bright and enthusiastic people to collaborate with, and it was really conversations with George that have sort of kept us going through the years. So I will talk about that microRNA story that you can kind of see here in the schematic in the lower right-hand corner here. But we also work in a couple of other areas looking at targeted therapies for head and neck squamous cell carcinoma in general, and also thyroid cancer, and specifically anaplastic thyroid cancer, and working through some of the rare tumor initiative with the institution, and then starting the FAST program with some of my clinical colleagues in order to really focus on anaplastic thyroid cancer. And I know last week, Cullen Taniguchi was in to talk about sort of normal tissue toxicity or toxicities related to radiation therapy. And that's been an area of interest for us too, as we've gone to look at sort of treatment effects of radiation in this sort of center grid, looking at some of the effects of radiation treatment on the mandible and causing osteoradionecrosis. And then here on the left-hand side, you're seeing a little GIF in the lower left-hand corner of pyruvate to lactate actually being converted inside an orthotopic model of anaplastic thyroid cancer. And as that's happening, you can actually see the last step of glycolysis happening, especially in tumors like head and neck cancer and anaplastic thyroid cancer, where the Warburg effect and aerobic glycolysis is critical. And the happiest part of that, and part of why I do this as a clinician scientist, is I show you this model here today. It's an orthotopic model in a mouse. And in late November, early December, we were actually able to do this finally in humans here at MD Anderson, and to do it in thyroid cancer patients as first in human ever in the world. And so we were very proud of sort of achieving those translational steps to make that a reality. But with that, I really want to focus today on head and neck squamous cell carcinomas, or oral cavity squamous cell carcinomas. And I show you some of the work from our labs here at MD Anderson, in collaboration, of course, with a lot of other labs, but looking at some of the sequencing data that was generated from human specimens, and looking at sort of signaling pathways that seem to be critical, or common molecular drivers in oral cavity cancer. I show you this as a background because you're going to see a lot of these pathways come up as we start talking about miR-27A star and sort of its regulatory role in HNSCC. And so, of course, things that you would expect to see, the mitogenic signaling pathways, you can see EGFR, PI3 kinase, acting then to AKT1, and then you'll see later the involvement of mTOR. And then, of course, the P53 pathway that's very commonly mutated, as you can kind of see down here, the four sort of genes that are highlighted here, the very often sort of mutated P53, the sort of related family member P63, and then to a lesser degree, but definitely also with some degree of mutation or copy number alterations, EGFR, the epidermal growth factor receptor, and then AKT1. And I show you this, and I'm sure many of you are already familiar with this. This is by no means meant to be sort of a comprehensive review of this, but you know, the P53 family, of course, is quite complex. It has a P63 sort of family member, of which there are multiple isoforms transcribed from at least two different promoters. The TA P63 has this sort of P53-like transcriptional activation domain. And then the other one that I'll sort of highlight because it comes into sort of the story that I'm telling you today is the delta N P63, which does not have the N terminus, or the transactivation domain at that N terminus. But it has been shown in a very nice story told by Jim Rocco and his group at Ohio State that delta N P63 is overexpressed in HNSCC and does promote its survival. So I go back a little bit of a ways to the initial starting point of this story, and sort of maybe the dogged pursuit of a particular sort of angle at understanding head and neck squamous cell carcinoma. So certainly some of the efforts with all the new sort of genomics and different omics platforms has been to try to identify new targets. What's always been intriguing to me is the epidermal growth factor receptor, or EGFR. The basis quite some time ago was that there was frequently overexpressed in head and neck cancer. So in tumors and in cell lines, over 70% or so of these cell lines and tumors demonstrated there was overexpression. And that increased expression was correlated with sort of decreased survival for these tumors. And so there was a lot of preclinical work done targeting EGFR and demonstrating that, in fact, direct targeting of that receptor seemed to inhibit tumor growth. And so cetuximab, which was had a monoclonal sort of antibody end to it, but was directly targeting here, as you can see in the figure, the EGFR receptor, was actually approved in 2006 for the use against head and neck squamous cell carcinoma in combination with radiation therapy. Now, there were also small molecule TKIs or tyrosine kinase inhibitors that were also approved or that were also being investigated. Now, interestingly, while the story from a preclinical standpoint and its sort of development made a lot of sort of scientific sense, what has not sort of borne out in head and neck squamous cell carcinoma is that alterations of expression level and signaling activity have not necessarily with treatment response. So you may see a tumor sort of respond or a patient respond to a particular therapy, to cetuximab, but you weren't always seeing a decrease in sort of receptor expression level or the downstream signaling events as a result. And so that was all sort of puzzling as we were sort of putting this story together. And it was around that time that I was sort of learning more about microRNA and microRNA processing, and we were starting to look at the issues of RNA, and so of microRNA and its activity and its ability to cause these to build these sort of silencing complexes, and to understand how the microRNA sort of was transcribed as a pre-microRNA and then turned into a duplex, and then that duplex was separated into into two strands, and then could cause either target inhibition inside these silencing complexes, or could actually lead to degradation of the messenger RNA. And so I start with this story by telling you that what we did is we looked at an initial exploratory question, and we said, you know, if EGFR is sort of our initial platform, can we identify microRNAs that may alter the activity of this signaling axis? Was the act of targeting EGFR itself not sufficient, and we needed to sort of have more targets along that entire axis? And in fact, of course, because of the way microRNAs sort of can target multiple sequences and have sort of evolved to have sort of a complex regulatory sort of program, the microRNAs seem to be an interesting place to start looking. And so, you know, we started to look at these sort of issues. It was sort of interesting to us to kind of understand that some microRNAs, you know, there were initial papers that were being published about microRNA profiles in head and neck cancers, some with low expression levels, some with high expression levels, as compared to sort of normal cell lines, epithelial cell lines and things like that. And so when we started looking, what we did is in silico, we did this analysis where we sort of looked at microRNA potential targets inside the EGFR gene, and inside, of course, its 5' UTR, but also inside the 3, and of course inside the 3' UTR. And what we found were a number of targets here, and a number of them, actually, I think MIR7 and MIR27A had already been reported as downregulating EGFR. But it was my initial visits here at MD Anderson and a conversation with George, where George had said to me, hey, you might want to be looking at this sort of star sequence, because here that maybe we haven't been appreciating about microRNA. And so by way of background, I show you this. The interesting thing is, of course, I showed you that duplex. And the duplex, it had sort of been assumed that one strand was kind of the active strand, and the second strand was kind of the lagging strand, or the star strand. And that while it was necessary to kind of create the hairpin and then to create the duplex, that star sequence probably didn't have much functional activity. And so, in fact, it was just thought to be a byproduct of, in fact, the MIR27A production, and so that MIR27A star would just be sort of degraded. Well, I think one of the most interesting things that we sort of saw was that, you know, and to some degree it was degraded, and in head and neck cancer, interestingly, it was very it was suppressed to quite a large degree, as you can see here. But and then you can also see that furthermore in some of the cell lines. So we are blessed in head and neck cancer, unlike some other sort of cancer types, in that we have actually several hundred different cell lines that we can use looking at sort of human HNSCC. So there are a lot of cell lines that we can use, and in fact what you can see here is the relative expression to sort of fibroblast or keratinocyte cell lines was that there seemed to be quite a bit of suppression of both the miR27A as well as the 27A star in those cell lines. And we could see that this was also differentially expressed in squamous cell carcinomas using, and now in the broader locus of the miR23A, 24, 2, and 27A locus, that it was sort of differentially expressed in squamous cell carcinoma. And so what was interesting to us then was, as we sort of took this initial panel and then we started to express the microRNAs using mimics with 27A, 27A star, and 7, is that in fact what you could see is that there was downregulation with the 27A star of EGFR, and you could see that also 7 and 27A had some downregulation also. But then it was furthermore that it was really 27A star that then seemed to also act sort of most impressively against both AKT1 and mTOR downstream. And that had also been sort of borne out by some of our initial in silico analysis. And on top of that, not only, so what we found was that the 27A and 7 would downregulate EGFR, were not acting to downregulate downstream of EGFR, and that in fact when 27A star did act downstream of EGFR, that in fact it also looked like it was causing increased apoptosis in these cells, and you can see that here. In our flow cytometry experiments, as well as in sort of cleavage of PARP, as demonstrated here below, again in a sort of panel of cell lines. And you could see also that when we introduced the miR27A star versus sort of other family members within that locus, while you could see downregulation of EGFR to sort of variable degrees with some of the other family members, again, it was really the difference here in sort of decreased cell growth and in fact increased apoptosis that seemed to be most profound with the miR27A star as compared to other sort of related microRNA in that locus. And I show you this. This is using a InQSite, and I like it because you can actually watch over time as these cells are growing. And I'm trying to see if we can get both videos to run simultaneously. But over the period of almost 2 days, as you can see, you know, with a control microRNA, the plate of cells is becoming somewhat confluent. Here you can see there's still quite a bit of space, and then there are sort of these very bright pycnotic cells that are indicating apoptosis is occurring. And then the summary slide here, where you can see the continued sort of increased level of proliferation inside that InQSite assay with the control microRNA treatment versus the miR27A star. Let's see here. Okay, good. And so I show you this again just to kind of remind you, and you know, we went back then to kind of human specimens to kind of look and to verify that in fact there seemed to be some active suppression of miR27A star, and of course in controlling that locus also miR27A. But it really again seemed to be miR27A star that was most active in sort of our laboratory experiments against head and neck HNSCC sort of tumor growth. And similarly, I tell you this story, and for those of you familiar with sort of head and neck, you know, one of the big sort of changing factors over the last decade or plus for head and neck cancers has been the involvement of the human papillomavirus. And so certainly HPV positive disease is more virally driven than sort of by oncogenic mutations and changes, although that does have some involvement. and sort of the traditional sort of tobacco and alcohol sort of contributing factors. But HPV positive disease, we also found similarly that sort of higher expression of the microRNA, where it was being less actively suppressed, those patients seem to do better than patients with sort of low levels of microRNA. And similarly, I show you this as sort of a graphical sort of display of those patients with slightly higher levels of miR27A star in the HPV positive. As similarly, as tumors sort of advance, there seems to be more active suppression of miR27A star, as you can see by AJCC staging of tumors, levels 1 and 2 versus levels 3 and 4, or stages 3 and 4. And so we had kind of gotten to this the story of miR27A star. It actually actively targeted EGFR, AKT1, mTOR, so sort of down the signaling pathway. The interesting sort of biological sort of story that developed out of this from sort of a mechanism standpoint was sort of understanding that it was not just the lead strand that was critical in sort of microRNA biology, but in fact what had previously been called the passenger strand or the star strand could actually also have biological activity. And so there have been other sort of star strands subsequently that have been demonstrated to have activity. But the other sort of interesting finding as we were sort of looking at this was that in fact head and neck squamous cell carcinoma and other tumors actively repressed expression of miR27A star, and that seemed, at least in our working model, to be useful for survival of the actual cancer. And so we started to work on this, and you know, of course, I already mentioned Nikhil Chari at the start of this, but he certainly gets the lion's share of credit for sort of driving this forward. But we wanted to understand what was sort of controlling then miR27A star expression. And, you know, as we sort of started looking at the locus, you know, we started thinking about the biology of head and neck cancer, and I mentioned to you already the important role of the p53 family. We started looking at this notion that delta NP63 alpha may actually be critical. And so I show you a number of experiments here and years of work by Nikhil that was recently published then in the JNCI, but using delta NP63 siRNA to target delta NP63 in a variety of head and neck cell lines, and you can see labeled here at the bottom. We sort of label the status of it, whether it's p53 null, p53 mutant, or p53 wild type. You can see the delta NP63 when it's being provided, then in this sort of bottom row of graphs, then the relative level of delta NP63, and also here in the immunoblots. But what we saw is when you did introduce delta NP63 siRNA, that in fact you also knocked down the miR27A star expression levels across all of these in sort of a different p53 backgrounds. And so that was sort of our initial sort of foray into looking at that issue. Now, one thing with the delta NP63, as you use that siRNA, and the data is not shown here, but we saw that TAP63 would actually, those levels would rise. And so there was a question of, was this a downregulation of delta NP63 or an upregulation of the TAP63? And in fact, when you do downregulate all forms of P63, you do still see a similar effect, even though there isn't as significant an effect on TP63, but it did seem to be that it was the delta NP63 that really was causing this difference. We went on to then, or Nikhil really went on and sort of drove this, looking at the miR27A star promoter by chip. Was able to, identify a couple of promoter, the regions within the promoter of this area that seemed to be critical for miR27A star. Further on, sorry, let me make sure we're heading up. There we go. Okay. What he's able to do is first, of course, using sort of a luciferase a promoter luciferase sort of construct demonstrate that introduction of delta NP63 did really sort of drive this forward using a luciferase assay. And then sort of building the mutant and the wild type and mutant promoter regions to really demonstrate that when you then did downregulate delta TP63, that in fact you could affect then the luciferase activity and sort of confirming this actual promoter site. And so here in the lower left-hand corner, right-hand corner, I just show you that sort of initial finding or the initial schematic that delta NP63 does seem to drive miR27A star expression. All right. So as I sort of mentioned to you, a number of the sort of findings that we were kind of correlating sort of laboratory findings with what we could obtain from human samples, and a lot of this was done through work with George and Christina Ivan, and so we're very appreciative of that. But the sort of first sort of gem in this sort of regulatory pathway was that delta NP63 was transcriptionally regulating miR27A star expression. This is of course complicated by that inside head and neck squamous cell carcinoma, that of course mutant TP53 also P53 is mutated quite often, almost over 70% of the time, by head and neck. By P53 mutations occur quite often in HNSCC, and so what we started then to look at is if you could downregulate P53 and its mutational forms or things like that, or if you had it present in some of these null cell lines like the SCC1 and PCI13, if you then reintroduced activating P53 mutations, what would happen then to the miR27A star levels? And in fact, what we saw is that if you used an siRNA against a mutant form of P53, as shown in this HN31 cell line, in fact, miR27A star levels went up. With the presence of the activating mutations in these two cell lines, in fact, miR27A star went down. So that there seemed to be a sort of a repression of the miR27A star expression by P53 mutants. And we further confirmed this. Carlos Cowlin, who was previously here at MD Anderson, he had murine models of P53 as mutations or null, null expression. But as those mutants were sort of being reintroduced into these models and cutaneous squamous cell carcinomas were developed, you could see that in fact there was sort of a relative suppression of the miR27A star expression levels in those tumors. And so again, when you sort of look at this is a system that was described through the Myers lab with looking at P53 scoring, sort of low risk versus high risk, that with the higher risk or more active mutations for P53, that in fact there seemed to be a relative suppression of miR27A star levels in those tumors compared to kind of wild type or low risk mutations. And so I show you that. We sort of further broke it out into oral cavity cancer and saw the sort of relationship, although with smaller numbers, was also present. And so this was a phenomenon that we could describe that seemed to be occurring both in HPV positive as well as in HPV negative disease, oral pharynx disease or sort of back of the throat and tonsils being more related to HPV positive disease, anterior kind of oral cavity and being more HPV negative forms of head and neck cancer. And so we started then to kind of look at the other direction, is then what was miR27A star doing to this sort of transcriptional pathway. And so what was done was we developed a system where you could use doxycycline to actually inducibly express miR27A star. And so again, Nikhil doing a lot of this work, but and Shangdong Li helping with some of the construction of the various plasmids and constructs to allow for this. But in fact, what we could show is that with doxycycline, you could get expression of miR27A star. You can see here on the upper right hand corner, expression of miR27A star increases with the doxycycline in our engineered cells. Again, it's very specific. You know, I talk about the hairpin loop and the sort of shared duplex for microRNA processing in sort of normal biology, but then with this, just showing that miR27A star is increased, but miR27A is not. And then subsequently, with that induction of miR27A star, again, holding on to demonstrating that in both an MTT assay as well as a clonogenic assay, that in fact you get cell killing or reduced viability in those cell lines. Now, so this is kind of the feedback mechanism, and I sort of show again in that lower right-hand corner the schematic. So the initial work to show that the delta NP63 was acting on the promoter of the miR27A star locus or family to increase its production. Similarly, and with some of the work that we had done earlier with EGFR and AKT1 and mTOR, we were then able to demonstrate that in fact delta NP63 is also a target of miR27A star. So adding to kind of that family or that sort of program that miR27A star can sort of regulate inside cells. And so on the left-hand side, you see here as you activate the miR27A star expression with the doxycycline, that in fact you got downregulation of delta NP63 expression in the cell line that we're working with. And then the sort of experiments where you have the luciferase assay with the 3' UTR sort of binding elements for miR27A star. You can see where the predicted binding sequence is in that 3' UTR, sort of then in its wild type form, and then once we mutated that area to demonstrate that you sort of where in the wild type you would get repression of luciferase activity, that then it was abrogated by the mutation, demonstrating that the 3' UTR was sensitive to regulation by the miR27A star. So, you know, of course, you demonstrate that it sort of affects delta NP63 alpha regulation, and then you go on further to show that downstream to that, of course, then it affected sort of expression levels of the mRNA that were known targets of delta NP63, including Noxa and Puma, with the upregulation of the miR27A star. And so sort of building again on that schematic, sort of the feedback loop here that you have between miR27A star and delta NP63, and sort of both of them regulating each other. The complexity of that, of course, being modulated by mutant P53 status, and then that all acting sort of downstream then to affect head and neck sort of squamous cell carcinoma growth and overall survival. I'm going to show you sort of as we, I want to make sure we have some time for questions and things like that. But, you know, as we sort of look further on, we're kind of interested in what are the other targets and sort of what are the other sort of elements that may be critical in this pathway. I show you NUP62 here. It's overexpressed in squamous cell carcinoma. It does definitely mediate transport of delta NP63 to the nucleus. And depletion of delta NP63 in squamous cell carcinoma, of NUP62 in squamous cell carcinoma can inhibit proliferation and induce differentiation. And we're able to show, again, sort of slightly increased expression here of NUP62 in head and neck cancers. And then, again, starting some of our work here, initially a small change in mRNA. And again, not wholly surprising if you sequester the mRNA and don't actually degrade it, but a much more convincing signal here with the decrease of the protein expression. And then similarly, initial studies that have demonstrated again, with the luciferase and the 3' UTR, that in fact NUP62 does seem to be regulated by miR27A star. And so again, adding now to that sort of diagram that I've been sort of building here in the lower right-hand corner of the sort of regulatory pathway that we're looking at with miR27, A star, and delta NP63. And so all of that sort of comes together now, and the work that we've been doing over the years, and certainly that I've been very grateful that George has sort of stayed in our corner and been enthusiastic about sort of pushing this forward and provide sort of ideas of how to sort of proceed with this. But, you know, we started with this notion that EGFR seems to be a very logical biological target in head and neck cancers, but the targeting of it alone does not seem to be sufficient. And so using then microRNA, we were able to find a microRNA here and sort of expand the biology in this area that not only did it target EGFR, but also AKT1 and mTOR. But then in understanding why it was being repressed in head and neck cancer, is sort of understanding that alterations not only in delta NP63, but also P53 as far as its mutational forms, seem to also have an important effect on sort of levels of miR27A star and then its overall activity or its lack of activity per se in squamous cell carcinoma, and then how that affected head and neck squamous cell carcinoma growth and survival. And then sort of trying to now understand and seeing, of course, that NUP62 is an attractive target here. But again, as it's an attractive target to miR27A star, also seeing how it's intimately involved with sort of the transportation or transport of delta NP63 to the appropriate sort of location inside the cell for its activity. I think as we sort of understand this biology, we also have come to understand that microRNAs represent sort of very elegant regulation inside normal cells, inside head and neck, inside cancer cells, and so one of the things that, of course, to try to bring this to reality is, can we actually deliver the microRNA to the appropriate target? And so we've done quite a bit of work. I show you some work that we did here initially, looking at these small lipid nanoparticles as a way to try to deliver it. Some of these aptamers, some of these nanoparticles, we tried to use aptamers like the AXL receptor as a way to sort of deliver into cells or cell lines. And you can see that with that here, we were able to deliver and sort of downregulate EGFR and AKT1 in some preliminary studies here with the SLNP. I show you this. This is work that we've been doing more recently with Lisa Villanueva at the University of Pittsburgh. She's a cardiologist. She's actually shown that delivery into cardiac muscle of microRNA, and has been using this also in head and neck to try to deliver some STAT3 or anti-STAT3 decoys. And so it seemed like potentially an attractive model here to use for delivery of head and neck squamous cell carcinoma, especially if we were interested in something like the oral cavity. What you can do is the microRNA or sort of your payload is put into microbubbles, and these microbubbles then you can actually image with the ultrasound as they're sort of being delivered into the tumor micro circulation. And when you pulse the microbubbles with the ultrasound signal, you can actually then cause the microbubbles to be destroyed, presumably then delivering their payload into the tumors. And then and so I like these images because you can see sort of the recycling as the tumor is filled with microbubbles. The ultrasound then allows those to burst, and then the microbubbles refill into the tumor, and again can be activated in a very sort of localized way, rather than sort of systemically sort of being burst into sort of normal tissues and other structures. So I show you this as the SCC7 is a murine squamous cell carcinoma model, and we've just been able to do some experiments with an EGFR reporter, so a luciferase with a 3' UTR from EGFR, simply to demonstrate that in vitro, when you deliver the microRNA, so control with the ultrasound versus the microRNA with the ultrasound, then in fact the ultrasound sort of activated microbubbles seem to be delivering the miR27A star into these murine squamous cell carcinomas. The microbubble delivery, initially the luciferase assay, and then as well here with the Western demonstrates that we're actually decreasing the level of expression of EGFR inside those cells. And then sort of a larger scale experiment demonstrating let's see here. Moving from the initial sort of in vitro studies to then an in vivo study where we created an orthotopic tumor inside the animal's tongue of a cell line, and then actually delivering microRNA either free systemically or then infusing it packaged inside the microbubble, and then using the ultrasound to activate it. But we could demonstrate, and you can see quantitation here on the right-hand side, but you could actually demonstrate that we were downregulating both EGFR, AKT, and mTOR in the animal. And then furthermore, in the P63 pathway, I think extremely clearly here with delta NP63, and still significantly here with NUP62. But again, with the ultrasound, and rather than just and packaged in the microbubble rather than the free miR27A star, you could see good downregulation of the expression of both of those targets. And so that's some of the work that we've been doing in that area. The last area that I'm going to be touching on and that we're continuing to work on is, understanding how to sort of, so one, can we deliver the microRNA? And then two, sort of understanding with the multiple pathways and alterations and sort of complexity of head and neck cancers, can we deliver useful therapy in combination with other available items, other available sort of targeted therapies or systemic therapies? I show you this because this was inhibition of EGFR with miR27A star and with erlotinib. Now, the interesting thing about erlotinib is, as I showed you at the beginning, erlotinib is kind of the cousin of cetuximab. Cetuximab has a monoclonal antibody sort of common chain at the end of it, and has now been sort of associated with some of the immune response as part of its effectiveness. Erlotinib is a small molecule TKI. Interestingly, because in head and neck cancer, EGFR is often overexpressed, but not as often mutated. In fact, erlotinib seems to have much less activity in head and neck cancer in patients than, say, in lung cancer, where a mutation found specifically in EGFR seems to be an indicator that erlotinib will have very good effect there. So we have not had a lot of success with erlotinib alone. Now, interestingly, erlotinib does show in vitro effects, and so I show you this as an example of sort of combinatorial therapy, where we're targeting both miR27A star as well as we're using both miR27A star and erlotinib to sort of target the same pathway. And in fact, what you see here is that both of them together have sort of similar effect. This is in an orthotopic mouse, again, tongue model. But then when you then put the two together, you seem to have even more significant effect. I'm going to call it additive at this point than either compound alone. And so we've been working with Clifford Steffen at the part of the Gulf Coast Consortium at the Texas A&M ITB, and been looking at sort of doing high throughput screens, looking at combinations of drugs and available compounds through his various libraries in our cell induced the miR-27 star. And so I show you sort of these tree analyses that come out, and we've identified sort of emetic regulators as well as kinase inhibitors that seem to act well in combination and may provide ability to sort of work together with miR-27A star. Some of the work that we're also doing as we're sort of developing this, one of the things that they can do over there is look at organoid cultures. So we're organoid cultures seem to be useful for assessing drug effects, especially in these sort of three-dimensional models. So we're developing some of those models also with them in an effort to kind of identify potential combinatorial effects that'll be useful. And so with that, I really do appreciate your time. I hope you have come away with a little bit more knowledge about head and neck cancer with microRNAs in general, and then understanding what we're trying to do in terms of improve treatment for our patients. Again, I want to highlight my group. I miss seeing them on a daily basis. The distance of the pandemic has been isolating. But I do appreciate efforts and opportunities like this to be able to communicate with our MD Anderson community and with our colleagues around the world. I want to again sort of emphasize a lot of this work has been driven by Nikhil Chari. I'm very appreciative of that. I also want to thank our collaborators, people from the Myers lab, the Pickering lab, our group that's been working with us now from Pittsburgh in the Villanueva lab. And then I mentioned George already and Christina Ivan for her fantastic work, and the others who have been in our group and have helped us in the various aspects of our study. And so with that, I appreciate your time. I appreciate your attention. Thank you very much.
Thank you very much, Stefan. It was a wonderful presentation. Of course, we love it because we love microRNAs and non-coding RNAs. And let's start with the question. So the first question. There are evidence in breast cancer that EGFR signaling can regulate microRNA expression, upregulating, downregulating. Have you checked if EGFR can regulate microRNAs also in head and neck squamous cell carcinoma? There might be either positive or negative feedback regulatory loops through EGFR and microRNAs.
Yeah, and so we've definitely been looking at those kinds of issues. You know, as you sort of look at the various pathways and various overexpression levels of EGFR, we sort of looked at that relative to miR-27A and miR-27A star. I think it's interesting, the question is certainly interesting in terms of looking more broadly across the profile of microRNAs. And so that might be an area that we'd want to kind of look at more broadly. So thank you.
Thank you. Another question. So how do you envision the continuation of your studies on the combinatorial therapy microRNAs plus another drug? Are you looking also for checkpoint immunotherapy combination? And so the question is about the continuation of the therapeutic study.
Yeah. So, you know, I think, of course, one of the things that you're always, you have to understand the limitations of the models you're working with. And so as we sort of do this in sort of the hybrid sort of human cancer in an immunocompromised animal model, you don't get sort of the full extent of the sort of disease spectrum and sort of the immunological response. And so that's partly why we've done some work in sort of murine setting to be able to look at sort of more of the immunological response. And so I think as we do the screening, we're certainly interested in checkpoint inhibitors. We're interested in those kinds of possibilities, but we need to develop better models to sort of fully understand those potential effects. And so we're definitely looking at doing those sorts of things.
And now one question from myself. So are you checking also on non-coding DNA mutations in this model? So we speak about non-coding RNA, but a lot of emphasis is lost from the fact that the genome has DNA non-coding sequences who do not produce RNA, but they are important, insulators, enhancers, and so on. Are you also focusing on this type of research?
We haven't done a lot of that, and I think that's an interesting point. I think the dark matter in the genome, I think is absolutely fascinating. So George, I think you and I will probably be having more conversations in that regard, but I think that's an absolutely fascinating area to look at. You know, we talk about omics and we talk about more and more sort of mapping of genetic information, and yet I think sometimes we forget just how small a sort of candle we have actually looking at sort of the entire sort of genetic program or genomic program that's going on inside even normal cells. So I think that would be a fascinating place to look. It certainly was the impetus for what we did here. And biology team solving for EGFR, its overexpression in head and neck cancer. Certainly have been efforts since to kind of look for the new sort of pathways like Notch and sort of other sort of screening elements like aurora kinases and things like that as they've sort of come up. But I think I still hold to the interest that this biology of the EGFR signaling pathway has been important or has been recognized to be important in head and neck cancers. And I think there's still a very sort of compelling story for actual therapeutic sort of targeting in that domain.
Now you said a magic word. You went a little outside to this new therapeutic, and in fact, it's a new question considering a high degree of tumor heterogeneity and deep intrinsic resistance. Are there any broadly active agents that have promised? So outside microRNA, I'm understanding. What are the agents that you are using for tumor heterogeneity?
And that's a really interesting question. And for as much as we've developed new targeting and talked about individualizing and personalizing therapy, I think one of the big studies that was recently completed by one of the cooperative groups, RTOG 1016, there had been a hope that we could set a new standard by which then you could compare immunotherapy. And we keep going back to cisplatin. Cisplatin, cisplatin is no longer sexy, but cisplatin is profoundly effective, and so in fact, in head and neck cancer, one of the big questions that's coming up soon in the cooperative group that they're going to spend a lot of time and effort and enroll potentially over 1,000 patients to answer this question is, over time, there has been an effort to change the weekly cisplatin sort of regimen during radiation, so concurrent radiation and chemotherapy. Cisplatin had been given on a weekly basis. And now some places say we're going to give it every 3 weeks. Okay. And by giving it every 3 weeks, hopefully we can reduce the toxicity, or, one or the other is going to reduce toxicity. Is it a weekly dose at weekly at a lower dose, or every 3 weeks at a higher bolus? It's fascinating to me that even now chemotherapy in that sort of concurrent radiation setting is being driven by toxicity, and toxicity to normal tissue, not actually toxicity to the cancer cells. And so I don't even have time to get into that issue, but it's interesting you bring up heterogeneity, because in fact, one of the things we think is absolutely critical about concurrent chemoradiation therapy is that you have to time when you give the chemotherapy or the cisplatin to actually optimize the radio sensitization of that chemotherapy. And so we don't even have time to get into that today, but I think that also then contributes to this notion of personalization and to sort of dealing with tumor heterogeneity and understanding the relative resistance of different portions of the tumor, not only to the chemotherapy, but to the radiation. But it is interesting as we keep looking for new pathways and new targets and things like that, that chemotherapy like cisplatin has been so effective and just an absolute mainstay in the armamentarium. So sorry, a little bit long-winded on that explanation, but
I think it's very important because you bring to the attention of all of us the fact that so-called all therapeutics still have great promises, and you have to use them in a new way in order to boost therapeutics. So not everything what is old is not useful. Old means sometimes also very useful.
Yes,
This is the message from your answer.
absolutely.
With this, thank you very much, Stephen. It was wonderful talk, and in fact, we all learn not only about microRNA, but about how to think therapeutically on the non-coding RNA field. It was a wonderful talk, and I want to thank to all of you being here with us. And in fact, in 2 weeks from now, again, we have another very interesting talk by Georgios Karas, a talk on heat shock protein 90. And with this, I wish all of you be happy, enjoy science, enjoy making discoveries. And I am looking forward to see you in person, because certainly in the near future, we will see each other in person and we will stay closer and closer, because science is all about communication. Thank you again, Stephen. Thank you to all of you being here with us. And thank you, Liz. Thank you, Laura. Good luck to all.